Hello and welcome to another lecture. This lecture is about flower induction. So last time we talked about the genes that pattern the flower once the flower is induced. So once the flower knows that it starts to flower, it's making the right organs and it patterns them in the right way, in the right whorls, and with the right number of petals, sepals, stamens, and carpels. Today's lecture is about the question, how does a flower know, how does a plant know when to make a flower and when to turn these genes on that eventually pattern the flower before it opens? So many of the pathways that are involved in flower induction, and there are numerous of them, have something to do with timing, with light and with the timing of the year when it's time for the plant to flower. So we talked a little bit about circadian rhythms before, and I just want to spend a little bit of time here at the beginning going over some nomenclature, some of the terms that are part of this circadian rhythms biology. So circadian rhythms are simply responses that happen in a periodic way. So for example, every 24 hours, once a day. So you can see here that the terminology that is used is the amplitude, that is the strength of the response. And then there's the period, that's the time that is between two events within a circadian rhythmic period. So for example, 24 hours. And then there are these phase points. And the phase points are just simply events that happen over a certain period of time. So this could be, for example, things that happen when the light comes on in the morning. So if you have a light-induced change, so in this case you turn off the light, then what you can see is if you run this is a free running circadian rhythm at this point, because it's no longer entrained, it doesn't have any more the effect from the light every day to tell it what the 24 hours exactly are, then it changes in its amplitude. The time doesn't usually change, but it can change a little bit, so that's the period here, when it's free running. But when it is entrained by an impulse of light, then that impulse resets this period every time that impulse happens. So for example, if you have plants in a growth chamber and they're on a timer, and every 12 hours the light comes off and every 12 hours the light goes off, then you have the event of light on and light off being the event that entrains the clock to keep it at this certain period of 24 hours. And so if then you have a change in this impulse, so for example, you turn the light on and then you turn it off, and then you turn it on but not after the normal period of time of 12 hours, let's say, then you reset the pulse. With that you reset the clock, and with that you reset the phase. So this would now be a phase change, but it would not be a period change, right? Because the period is the same length. It's just the phase that has changed, the events that occur when the light comes on. I have on the next slide a summary of these terms, but I wanted to just show you why that is of importance, because the circadian rhythms, of course, change over the year in terms of the impulses that reset the clock. So over the length of over the area of the Earth, you can see that the length of the time of daylight varies, of course, with the time of the year. And so here you see just some examples of how long a day, so from sunrise to sunset, is at the various degrees of latitude. So the 60-degree latitude, so that would be somewhere around here, Stockholm, for example. Or Tacoma is at 47 degrees, so that's somewhere down in here in between. You can see the length of the day in the summer and the length of the day in the winter in January. Okay, so this is the terminology that I explained previously. A free-running circadian clock is in most species a 24-hour cycle in continuous dark or light, but this can change after a while. So you see that here, this free running clock changes after a time of no extra impulse. Light cues entrain the circadian clock and can shift the phase. So it can shift the phase here, but it cannot shift the period. The period is species specific. And these light cues are, of course, sensed in the plant via light receptors, such as phytochrome and cryptochrome. And then the term photoperiodism simply states or describes the ability of an organism to detect day length. Okay, so let's see how that has an effect on the induction of flowering, which is, of course, the topic of today. So photoperiod determines in plants flowering time, but it also determines formation of storage organs. So think, for example, about potatoes. In potatoes, the tubers are formed with response to the time of the year. And then dormancy of the seed is also affected by photoperiod, by the photoperiod. And in contrast, in animals, hibernation, so when it's time to go to sleep over the winter, the thickness of the coat or the color, for example, white coats in the winter, mottled or brown coats in the summer, and mating behavior are all photoperiodic events. Okay, so let's get back to the flowering as an example of a photoperiodic event. There are two major classes of plants that are called either long day plants or short day plants. And they can be longer day plants, if you will, and shorter day plants. So there's not just one particular time or number of hours that makes one species a long day plant versus a short day plant. So this can shift a little bit in this direction or in this direction. And what this is here is the percent of flowers starting to flower. So here you have no flowering at all, and here you have all the flowers on the plant starting to flower. And here's the day length, so how many hours of sunshine during the day, and the night length. And you'll see that the night length is just as important as the day length, and sometimes not the same thing. It's not just the reciprocal of the day length. You'll see that in a moment. So a long day plant, it flowers when the day length exceeds a certain critical duration. So what I mean here is that a certain critical duration in a 24-hour cycle is apparently here for this example of this long day plant, 11 hours or so. At that point, when that point is reached, all the flowers start to flower. So this would be a plant that waits in the spring patiently until the days are long enough so that it will then start to flower. And this is not directly dependent on temperature. So of course in the spring, the temperature also increases, right? But those are two different pathways. They're differently sensed. They do act together, but they are not directly related with each other. They're not dependent on each other. That's why you can have, flowering starts in the spring, even when you have a cold spring. Of course, in a warmer spring, that does move forward a little bit in the year, but it's not dependent on temperature exclusively. So even in a very cold spring, eventually plants will start to flower. So if we go to the short day plants, those are plants that flower in the fall. So they wait all summer long before the length of the day drops and reaches a critical number of hours. And so these short day plants flower when the day length is less than that particular critical number of hours. So here we have short day plants versus long day plants. So summary is that short day plants flower in short days or long nights, and that long day plants flower in long nights or long days or short nights. And I say here that the exact length is species dependent. So many species, they're not all flowering at the same time, obviously. So the exact timing of that species dependent. But you will see that at least for Arabidopsis and for rice, it's quite well known on the molecular level what determines flower initiation. And we'll get to that in a second. So I said that the night length is not necessarily reciprocal to the day length. So what that means is that that researchers have found that plants that are here, for example, a long day plant, that flower in long days. So here's a flowering plant, here's a 24-hour day, and the bar that is yellow is the time of the day, where the sun is up, and this is the night. So you see that in a long day, you have a long day plant flowering, and in a short day, you have a long day plant not flowering. But you will see that also if you have a relatively short day, same short day as here, but the night length has been interrupted by a night break, so that lights were turned on in this experiment for a certain period of time, that is enough to induce flowering. So the length of the day, therefore, right, this is the length of the day, or maybe you could say this is even the length of the day, that will be 2 days in a 24-hour time period. But the point here is that this is not important. What is important is that this is now, the length of the night is now no longer greater than the critical number of hours. And the same is true with the short day plants, where you have a long day and a short night not leading to flowering, and a long night leading to flowering in the fall. But when you interrupt the night with a flash of light or a night break, you can prevent the flowering as well. So night length must rise above a critical length. So we can say that long, short day plants, so long and then short day plants, they require long days before short days. And the short, long day plants, they require those are plants that require short days followed by long days. So spring versus fall. Okay, so question, of course, is how do we know what molecules, what genes are involved in eliciting these responses? So what is the molecular pathway for flower induction? How would you find that out? Well, of course, the answer for an Arabidopsis geneticist would be you do a mutant screen, right? So you look for mutants that are not responding normally with respect to flower formation. And then you look for the genes that are broken in these mutants, characterize those genes, and voila, you should have some information about the molecular pathway that is responsible for flower induction. But the question is, how would you set up such screens? So think a little bit about that. Maybe stop the lecture for a few minutes. Think about it. Write down a few ideas of how you would set up such mutant screen. We've talked about mutant screens at length in class. We've come up with some ideas for certain pathways. How would you set up a mutant screen here? Well, so if you thought about this, maybe you've come up with these responses or different responses. There are different possibilities of how you can do that. For example, you could grow a mutant population of a long day plant in long days and then screen for those plants that flower late. They don't flower when everybody else flowers, they flower later than that. So something must be missing in their response to the long days that doesn't allow them to flower immediately. Or you could grow a mutant population of long day plants in short days and then screen for those that flower early. So you could see what is it that allows these to flower early? Why are they not waiting for the flowering to occur when it normally should, even though the day is still short and therefore the signal hasn't been given to the rest of the population, the unmutated population, that it's not time to flower yet. So those types of mutant screens were done. And the example of growing a mutant population of a long day plant in long days and then screening for those that flower late turned up 2 mutants. And those 2 mutants were called constans and flowering locus T. And you see that there's a number of flowering loci. You will talk about some other genes that have flowering locus letter, but this one is flowering locus T. And so mutants that were called constans, they are late flowering in long days, and flowering locus T mutants are also late flowering in long days. So for these mutants, the genes were isolated through map-based cloning, same process that we discussed in class. And then the molecular identity of the proteins was analyzed using computer analysis to see if there were certain domains that had any similarity to other known genes, domains in other known genes. and then molecular analysis was done, gene-gene interaction, protein-protein interaction, protein-gene interaction, things like that, some of which we have discussed already as well in this class. In the end, what was found is that in long days, phytochrome A and cryptochrome 1 and 2 both were responsible for inducing constants, which was responsible for inducing flowering locus T, and that was then responsible for inducing flowering. But there's even more to know about the induction than just this linear pathway. So it turns out that the way that the plant determines the time of length, so the long days, how does it know long days, is more complicated than researchers originally thought. So it turns out that even with the knowledge of the constants gene and the FT gene, it took a while until people found out exactly how this works. So what happens in Arabidopsis is that in short days, the plant is vegetative, so it doesn't grow until the days become longer. And at that time, while the days are short, the constants mRNA cycles periodically, right? It goes up and it goes down. So at night, it goes up, and then when the light comes on, it gets degraded. But it turns out that the protein, the constants protein, doesn't actually change at all in its transcription. You would think that it cycles the same way as the mRNA, but it didn't. And actually, researchers didn't look for protein at the beginning. They just looked at RNA, and they were baffled because nothing happened. And that was because the constants protein was not translated. It wasn't translated until the day reached a critical length of hours. And at that time, constants protein cycled just like the mRNA did, but only when the days were long. So you see that here, night starts around here, and over here, night starts much later over here. And so the critical number of hours of transcription of constants mRNA was required for, at some point, several hours in, also acquire constants protein. And the reason for that is that FT mRNA, which is not transcribed, there's this little blip here, but it's essentially not transcribed in short days, is only transcribed in long days. And that is what allowed the constants protein to be made. So without the transcription of FT mRNA, constants protein was degraded as soon as it was made, and so therefore never reached a critical mass and never had its effect. So as a summary here of what I just said, in long day plants such as Arabidopsis, enough constants protein has to accumulate in the leaf to activate FT mRNA production. And constants protein is degraded in short days. And so you don't have enough FT mRNA if you don't have enough constants protein. In the long days, the cryptochrome and the phytochrome A, they sense the long days. The constants protein is not degraded. It activates the FT. It moves the leaf. The FT now moves from the leaf to the bud, and there it induces flowering. And we'll come back to this part of the pathway, how FT activates flowering in a little bit. But this is the interaction between constants and FT that is guided or regulated by light-sensing proteins such as phytochrome and cryptochrome. Now, how is that in the short day plants, you might ask? Well, it turns out that in short day plants, evolution didn't invent a whole different pathway. The only thing that it changed was that constants here does not activate FT, but it inhibits FT. So here's an example for rice, and over here on the side I put the Arabidopsis example just so that you have it side by side. And then I wrote up here that in rice, the names for the genes are different. They're called HD heading. So that's, when the flowering time in a grass, that's the head that is produced. So heading 1 and heading 3, HD1 is constants, HD3 is FT. So those are the homologs in rice to these Arabidopsis proteins. And so here you see that in rice, you have mRNA accumulating. You have HD1 mRNA accumulating at the same time as you have HD3 FT mRNA accumulating. That is different in Arabidopsis, right? So here it's the other way around. Here you have no FT or HD3 mRNA accumulating while the protein is active. So the point here is that Constans inhibits FT, and FT, when it's there, will induce flowering, right? But in Arabidopsis, Constans activated FT. Here in rice, it inhibits FT. So probably just a few changes in binding of proteins to each other that allow either activation or repression is what made the difference here. And so evolution did not need to invent a whole different pathway to sense short versus long days. Okay, so what happens, we're back to Arabidopsis here, when FT is activated. So what you have is light quality, day length, and temperature. And we now talked about how that eventually leads to the induction of FT. So you got that expressed, and that is happening in the leaves, right? So it's down here in the leaf. It's not in the flowers. FT is now expressed, and it's moved through the phloem, so through the companion cells into the sieve element, so it's part of the phloem here, and then it's moved up into the shoot apical meristem, so moves via the phloem into the SAM, and that's our step number 3 here. And step number 4 and 5 are then the interface of the flowering induction of FT and Constans with some of the genes that we have already heard about. And so the genes that we have already heard about that we discussed last time are the floral homeotic genes, right? Those are the genes that are patterning the flower, that are deciding what part of the apical meristem, of the floral apical meristem, are going to be sepals, petals, stamens, or carpels. But there's a set of genes here in between that activate these floral homeotic genes. So I want to spend a little bit of time discussing those with you. So you see here that FT protein is transported into the apical meristem, and there it interacts with another floral activation gene. So this is flowering locus D, flowering locus T, and flowering locus D. And when they bind, when they interact, they turn on two proteins, two genes. So they make two proteins via turning on the transcription of those genes. The left part here is the same as the right part. So just look at one part at a time. You see that this interaction here with FT turns on the transcription of a gene called SOC1 that's called suppressor of Constans. So when it was mutated, when they first found this gene, it was suppressing, the mutant was suppressing the action of Constans, right? Constans, FT moves up and then activates SOC1. And SOC1 then activates a gene that's called LEAFY, and LEAFY is the major regulator that regulates floral homeotic genes, so the ABC genes that we discussed before. And AP1, this is apetalor 1, this is not apetalor 2 or 3, this is a different gene with a similar phenotype, no petals, but it's a different gene from the floral homeotic genes we discussed last time. That is also upregulated by FT. So LEAFY and AP regulate each other, but together they then regulate the floral homeotic genes. And then you see that there's a number of other things that activate or deactivate SOC1. We'll come back to SOC1 as the central player in the determination of when flowers are made or when they are repressed. So let's just make a smaller, at this point still more simple diagram of the genes that are involved. So, sorry, we said SOC1 is a central player here, and day length activates SOC1. SOC1 activates LEAFY. leafy activates the ABC genes, and they then pattern the floral primordia, right? And this whole happens from a vegetative meristem going over to a floral meristem. So to get from vegetative to reproductive, day length will activate SOC1, which will activate leafy, which will activate ABC genes, and then that will make the floral primordia. Now, from what I just told you before, we now know that day length is sensed by phytochromes and cryptochromes. Constans activates FT via this mechanism of a critical time point, a critical threshold of time that is required for the accumulation or degradation of constans, which then leads to FT being either transcribed or not transcribed. When it's transcribed, it moves from the leaf to the shoot apical meristem. It activates SOC1 and it sets in motion this pathway. So there are a number of other inputs into this pathway. So we just talked about this part of the pathway, but we have in fact talked about other parts of the pathway already. We just haven't really mentioned it. So gibberellic acid, the hormone gibberellic acid, has many functions, and of course it has functions in dormancy and in shoot length regulation, but it also has a function in flowering. And it does that by interacting or activating SOC1 as well. So not only FT activates SOC1, but also gibberellic acid. Sucrose, so basically a sensor of the amount or the status of the metabolic status of the plant, will also activate SOC1. So that means a plant that is starved, that hasn't had enough time to photosynthesize, produce a lot of sucrose, and thus in the end support its floral primordia and the new embryo that it's making. If there isn't enough sucrose, then SOC1 is not activated, and so therefore flowering is delayed. Cold temperatures and an insufficient number of leaves, so that probably goes together with sucrose amount and status of the metabolites in the plant. Not enough leaves, not enough photosynthesis, not enough sucrose. So the insufficient number of leaves or cold temperatures will repress SOC1. So now I told you a little bit earlier in the lecture that these are independent pathways, and they are to some degree, but they all converge on SOC1. So this is the central player in the determination of whether or not the plant flowers. Now, here you have a day length sensor via constans and FT. Here you have a metabolic state sensor via sucrose and the number of leaves. Here you have an environmental sensor of the temperature that we'll discuss in a little more in detail. And here you have the hormone that helps the plant to grow and to develop. So all of those together then determine when SOC1 is activated. Now, we'll come back to this, but I think I want to make the point right here that, I talked earlier about particularly cold springs or very warm springs with early flowering. I said that even a cold spring, eventually the plants will flower. Well, that is because SOC1 has input from so many different parts of the sensory pathways, if you will. So let's say that it's a very cold year, and you have repression of SOC1 because of the cold. Well, you have activation when the days become long enough of SOC1, and at some point you have enough activation from this part, and this part to overcome cold temperatures. That just leads to late flowering. And that's great because late flowering means that the plant will reproduce. It will just reproduce a little bit later. And most likely that makes sense because it's no longer that cold as it was when the delay occurred. So all of these together will optimize the flowering time for the plant for a given year. We'll come back to that in a little bit as well. We'll come back to this particular pathway and we'll add on a little bit more detail as well. So cold temperatures, let's look at the molecular pathway of sensing of cold temperatures and how that interacts with SOC1. So of course, plant molecular biologists would employ their main the main trick, right? So they would do a mutant screen to find genes that are involved in cold sensing. And so what was done is Arabidopsis is a winter annual, or it can be a winter annual. There's some ecotypes that are. So you plant them in the fall, they overwinter as a seedling, and then they flower in the spring. And in winter annual, after 40 days of cold, with this particular gene that was found from the mutant screen that was called flowering locus C, you can see that this winter annual, after 40 days of cold treatment, does not express this FLC gene, the flowering locus C gene. No mRNA here. Here's the mutant, winter annual without cold, but with an FLC mutation, also doesn't have the FLC transcription. And here's what you have, a winter annual, what it looks like if you don't put it in the cold. So if you just leave it in the greenhouse or in the growth chamber, if it requires cold, it just won't start to flower. It will continue to make leaves and leaves and leaves and leaves until it reaches the time when it's time to flower. And it will, at some point, will be the time to flower, and that is because SOC1 might be activated by other pathways. But as long as the plant is not being treated with cold, FLC mRNA remained. So this mutant really was found that was flowering without cold treatment, without the necessity of cold treatment. And so the gene was cloned, the gene was characterized. Here you see the characterization of the transcriptional activity of the mutant versus the wild type versus a wild type that was treated with cold. And that seems to be part of the pathway regulating flower induction. And we'll fit it in a second. So let me just summarize this up here. Mutagenize winter annual cold requiring seed screen. So these are mutagenized seeds, of course, then screen for flowering without cold treatment. So those are these. And then clone the gene, measure the gene's RNA expression. And then you see that there isn't a gene here expressed and there is a gene here expressed. So what does that mean for the function? Well, it seems that as long as you have the gene there, that is not inducing flowering, right? Because this guy isn't flowering, and when you don't have the gene, the guy is flowering. So it must mean that the function of FLC is repression of flowering. So FLC is a major floral repressor. And when people looked at the gene and the protein structure and they compared it with other genes, they looked at certain conserved regions, and they found that FLC is a Matzbox gene. What else do we know about Matzbox genes? Well, we have talked about Matzbox genes in the context of the homeotic genes, right? The floral homeotic genes, many of which are Matzbox genes as well. So FLC is related to the genes that are patterning the flower, but it's a repressor of flowering time. So FLC is a Matzbox gene that inhibits downstream genes required for the transition from vegetative to flowering. So let's put that into our pathway here. So cold temperatures are repressing flowering locus C. And flowering locus C, when it is active, it represses SOC1. So remove the cold temperatures and you remove the repression of FLC, and you put in motion the pathway that leads from SOC1 via LEAFY and the ABC genes to the patterning of the flower. Okay, so now the question might be, well, how does that work? How does it, how is FLC activated or repressed? That is an interesting story, really. And one of the earlier, I won't say the first, but one of the earlier genes where epigenetic changes were found to play a major role in the transcriptional activation of these genes. So we're not talking about transcription factors that are turning on or off the gene, but we're talking about epigenetic factors. What are epigenetic factors? Well, epigenetic factors are factors that affect the gene on top, epi on top. So that means they're genetic in that genes are affected, but they're not, so you could change what's on top of the gene rather than what is in the gene. So that means the A, T, Cs, and Gs. If you change those, you will get a change in the activity of the gene, right, a mutation. But if you have an epigenetic change or an epigenetic mutation, what happens is that groups that are sitting on top of the DNA, oftentimes methyl groups, so CH3, that they activate or deactivate the transcription of this gene. I don't want to go into too much molecular detail on how that works, but let me show you this little cartoon here. So this thread is the DNA of the FLC gene, right? And DNA in the nucleus is wrapped around histones. Those are proteins that make sure that the gene is in a small bit of a, sort of wadded up. So it makes it compact. So histones compact the gene, otherwise it would be a very long thread, right? So this is spaghetti that is wrapped around a fork rather than just sort of randomly distributed on the plate. So this part here is the FLC gene, and these 4 dots, these circles, those are histone proteins. Histone proteins always come in 2s, and then actually they bind to other friends that they have, and soon enough there are 4, and then they're in duplicates of that, and then they're 8, and it doesn't matter so much. It just matters that these are histones, and the histones are proteins, and so therefore they are made of amino acids, right? Now, as it turns out, the amino acid strand that makes up these histones, these are the tails, these are the parts of the protein, of these histone proteins, that aren't wrapped up into these little balls. It's just sort of like a ball of yarn, and then you have a little bit of the end of the string hanging out. That is the histone tail. And of course, that yarn, that's all the amino acids strung together. And the tail then is made up of the remaining amino acids that are sort of just hanging off. Well, turns out that the status of the amino acids on these histone tails for the histones around which FLC is wrapped is very important. So you have amino acids, and every now and then you have an amino acid called lysine, right? Lysine is one of the 20 amino acids. And the way that they are numbered or named is that you have histones, H, and then you have these different types of histones. So you have a histone H1, H2, H3, H4, but the histone H3, that's the histone number 3, at the lysine K, that's the 1 amino acid abbreviation for lysine, number 9, counting from the end, 1, 2, 3, 4, 5, 6, 7, 8. This is 4 actually, and then this is 9. So when you count these up, the 9th amino acid from the end, if that's a lysine, then it's a K9, and if it's the 4th amino acid from the end, it's a 4, then that's K4. So that's just the nomenclature for it. Point is, what is the methylation state of these amino acids? So do they have an extra methyl group stuck on it or not? And if so, interestingly, that changes the activity of the transcription of this gene that is wrapped around the histones, which are modified. So you have histone modification of histones around which genes are wrapped, and the genes that are wrapped around these histones that are modified are then affected in their transcriptional activity by the status of these amino acids. So I wrote down here an H3K4 that's methylated is usually an active gene, and then H3K9 that's methylated usually is associated with an inactive gene. So if the FLC gene is wrapped around histones that have this methylation state, then they're active, and this methylation state, then they're inactive. Well, that is true for the most part, but there are exceptions, and it's not quite as black and white, and there are other lysines, and it turns out there are also other ways you can modify the histones, but we don't need to get into the details of that at this point. This is maybe what you want to remember as sort of a rule of thumb. And I just added here that this particular gene that's called VRN, or vernalization. Vernalization is means going through spring. Vernus in Latin means spring. So it really means going through winter and waiting for spring. It's a cold treatment when you call vernalization, when you say something is vernalized, you say that it went through winter. It was treated with coldness. So this gene, for reasons that will become clear a little bit later in the lecture, are called vernalizer genes, or vernalization. And this vernalization gene, VRN, deactivates FLC by methylating these histones. So it methylates these histones. Maybe it plucks away the active histones, the methylations at these sites that lead to activity. And that is the reason why FLC becomes either active or inactive. So how do we know this? I have to show you this part here. I'm not gonna go into too much detail, but let me show you this experiment that was done at the John Innes Center in England. Quite a groundbreaking experiment where they first showed how environmental cues, so temperature, can have an effect on the epigenetic status of genes, and how that then can affect their transcriptional, their phenotype, their transcriptional state. So let's say this is the FLC gene. This is the same gene that in the previous slide was wrapped around all these histones. Now the histones are gone. I'm not showing you the histones. I'm just showing you the promoter region. I'm showing you the exon. These are all exons. This is a longer exon, and it has a very long intron here. And then there's intron 2, 3, 4, 5, 6, and then there's intron 7. Anyway, exon-intron structure. And then what people did here is they wanted to see if the methylation of the histones that is associated with these particular regions here, these letters, A, B, C, D, E, F, G. So they looked for the methylation state in these regions that they called A, B, C, D, and so forth. They didn't look here, they didn't look here, they didn't look here. They just looked at where they point out the letters. And then these letters correspond with these letters. And so what they did is they did a certain technique. We won't go into the details of it right now, but essentially a PCR band in this assay here means that you have a methylated histone in that region. So if there's a band here in region A, so up here in the gene, there's a K4, so lysine 4 methylated, but the lysine number 9 is not methylated. So remember, these are numbered from the end of the tail of the histone that sort of hangs out of that blob. And the fourth amino acid in is a lysine, and that one is methylated here. But the lysine number 9 is not methylated at region A. Region B, it's now different. Region C is no different, and so forth and so forth. So in a non-vernalized plant, so a plant that has not been treated by winter, has not gone through winter, has just been sitting inside or in a greenhouse, you have the methylation state where K4s are methylated. K9s are not methylated. And when you put the plant through vernalization, you see that there are some changes. And the changes that you see is a removal of this activation mark in E, and an increase, an addition of a deactivation mark. K9's methylation at K9 is associated with deactivation of transcription. Methylation at K4 is associated with activation of transcription. So we're removing some activating marks and we're adding some deactivating marks. And now remember that this is a deactivating gene, right? This is FLC. So we want to repress it so that it stops repressing the flowering when it's time to go into flowering mode. So FLC is being deactivated epigenetically so that it will lift its repression of SOC1 and then allow the plant to go through to flowering. So vernalization leads to methylation of K9 and demethylation of K4. That's this here. That leads to inactivation of transcription of FLC, lifting of repression of downstream responses. So let me show you this here again. So these are the parts that we see changing. These parts are not changing, and apparently it doesn't matter. And apparently it does not matter that this is not methylated. Just these changes in regions A, B, and E are enough for the change in the transcriptional activity of FLC. And if we put this in the pathway that we've already started, we now see that VRN, vernalization, that gene, deactivates FLC via histone methylation. Now, when FLC is deactivated, it's no longer deactivating SOC1, right? So this is now no longer deactivating SOC1, and flowering can occur. And with that, you have multiple pathways from light through photoreceptors, temperature through epigenetic regulation of FLC, metabolic pathways that are indicating the metabolic state, and hormones, all converging on this major regulator SOC1 that together with all the inputs from all the different pathways will then determine when there's enough SOC1 activity that will lead to the activity of this intermediate leafy, and then the ABC floral homeotic genes that we discussed that will then lead to flowering. And that is what we currently know about how flowering is regulated molecularly. And with that, I will see you in class. Bye.